All right, this is the cell cycle overview that we did in class, and I just want to make sure that we all have this. I'm going to change the color of my pen here real quick. So we filled this in, but I want to kind of make sure we've got an understanding. So when we talk about the cell cycle, this is that important part, interphase. And again, you need to remember that these are the three parts of interphase: G1, S, and G2. G1 being growth 1, S, and then G2. So if we look, and then of course we also talked about G0, cells stop dividing, nerve cells will be in G0. But when I talk about interphase, make sure you understand there's G1, S, and G2. And so interphase basically represents the phases in between mitosis. So let me move this down a little bit. So we've got interphase. Let me go over interphase real quick. So I'm going to do interphase in, I think we'll go with green here. So, whoops. So if we go basically this pie chart here, and it's kind of hard to draw because I'm not looking at one thing. This is all interphase, and so there's G1, S, G2. G1, real way to remember when we're in interphase, we have chromatin. So in chromatin, so if this is my nucleus, the DNA is going to be all kind of unorganized. What you know about G1, cells doing its job, and its phase most of the time is G1, and actually it's interphase overall. So again, the key to interphase is this right here. Here's my nucleus. This is all my chromatin. S, just like what it sounds like, synthesis. We're making DNA. We'll talk about DNA replication here in a little bit. Excuse me. So that's the synthesis. And then G2, whoops, we're just having all kinds of problems today. Let me get this fixed real quick. I'm trying to get all fancy, it doesn't want to work out. So over here at G2 is our growth phase. Cells make molecules and organ needed for new cell. So again, from here to here is all interphase. All right, G1, S, G2. You should be able to tell me the three phases and what occurs in each. The one that we're going to be spending most of our time on is right here, mitosis. And I have this division sign here because this is where the nucleus divides. Bacteria don't do mitosis. They don't have a nucleus. So mitosis is actually division of the nucleus. And so if you say bacteria do mitosis, we're going to have a problem. But as you can see, mitosis, the cells spend the least amount of time in mitosis. But let's look down here at the different phases. And so when we talk about mitosis, we've got the four phases: prophase, metaphase, anaphase, telophase, cytokinesis. A lot of times when we talk about mitosis, we're only going to refer to these four, sometimes like PMAT: prophase, metaphase, anaphase, telophase. Cytokinesis, or between telophase and cytokinesis, indicates to most the end of the cell cycle. And so with cytokinesis, you should see two terms here: cyto, or you should be able to read it, and kinesis. Cyto refers to cytoplasm. Kinesis, a lot of times it deals with movement, but that's the division of the cytoplasm. So cytokinesis is division of the cytoplasm. Division of the cytoplasm. My writing's horrible today, but you get the idea. Now, I do want to go back up to G0 again. So when we talk about G0 here, again, no growth. Nerve cells. This is why nerve cells can't regenerate. They're stuck in G0. And so when we talk about cancer, we'll talk about G0 also, but this is the phase where cell stops dividing. And then what we're going to talk about later, in between some of these areas here, are checkpoints that kind of make sure that the cells get to divide. But I'm going to kind of go over that with you. The bottom part, which we've done, and you've already filled this in class, but I just want to kind of make sure you understand that, is we really got to have a good understanding of these different phases. So when we talk about interphase, again, in between the dividing, As you can see here, we've got a nucleus. The DNA is spread out like chromatin. You got to think of these proper vocab. So there's my chromatin. You've got a nuclear membrane. You've got the nucleolus, and DNA is copied. So if I change my color here, if it'll let me, my nucleus, I'll make it blue, or the nucleolus, cell membrane is still there. DNA is being copied, or what we call DNA replication. Prophase is the first dividing phase. Chromatin scrunches up and forms chromosomes. I don't know if you can see those little X's in there. They look like X's. Those are chromosomes. The nuclear membrane, it'll disappear. It's starting to break up. These things called spindle fibers appear. So if you look here, these blue things I'm kind of drawing over, spindle fibers, centrioles appear next to the nucleus and move towards poles. So let me do a different color. The white's not going to be good. Let's try green. There's your centrioles. And so prophase, a lot's going on because a lot of this new stuff is appearing. Meta is real simple. Middle. Chromosomes line up along the middle. So here's the middle of my cell. It's not a perfect line there. Let me redo that. It's going to be kind of tough trying to watch. Okay, so there's the middle. Again, not good, whatever. Chromosomes along the middle. All right, we've got our centrioles here. There's our spindle fiber or kinetochore. Key is they're in the middle because they're getting ready to divide. Excuse me. Anaphase, apart. The chromatid arm separates, moves to the opposite end of the cell. So if we've got a chromosome, pretend it looks like an X, there'll be a piece there in the middle. What will happen is it'll divide down the middle. This is a chromatid, half of it. So two chromatids equal one chromosome. And again, good luck reading that. Sorry about that. When we get to meiosis or meiosis, it's a little more complicated. But what you should be able to see here, got all this space. Because there's anaphase means that they're separating. Move on to telophase or telophase. Again, I'm sure, whoops, did not want to do that. Let me kind of go back. So let's go back, go down. We did anaphase. So telophase or telophase. We've got two nuclei. You're going to be able to see them both. Reverse prophase is sometimes what's called. Chromosomes spread out back as chromatin. Nuclear membrane returns. Centrioles disappear. So you can see why this is called reverse prophase. And so there's our nucleus coming back. Got some chromatin. But you're going to see this, you're going to start to see the cell pinch into two. So telophase, telophase, reverse prophase. Then our last is cytokinesis. The cytoplasm splits into two cells. If it's an animal cell, we call it a cleavage furrow, which is this right here. If it's a plant cell, it forms a cell plate, which is a nice kind of perfect little square almost. Cell plate will form here. What will happen is the Golgi bodies are what make the new cell wall in plants. Cleavage furrow just pinches in, but as you can see, there's our nucleus. We've got nucleolus. So again, we did this in class, but this was just to kind of give you a quick overview. Again, if you've got any questions, feel free to ask, but this is just showing you those two pictures that we did. So if you got any questions, feel free to ask, and yeah, good luck.